All right, let's get into it. You know that feeling, right? You open a scientific paper and just stare at it. Completely overwhelmed? Well, if that's you, you're in exactly the right place. Today, we're going to break down how to read, understand, and maybe even enjoy a research paper. We're turning that confusion into confidence. I guarantee you, every single scientist has had this exact thought. And I mean every scientist. It's totally universal. All that dense jargon, the complicated charts. It feels like you need some kind of secret decoder ring just to get past the first paragraph. But here's the secret. It's not about being a genius. It's about having a strategy. And speaking of strategy, let's talk about what happens when you try to take shortcuts. Picture this. Last year, in what's now famously called the Great Proposal Disaster, students let AI do all the heavy lifting for them. And the result? Ugh, it was pure chaos. We're talking cloned papers, totally ignored instructions, and get this, some proposals written in bold, size 16 Times New Roman. It's a pretty hilarious reminder that whether you're writing or reading, the real understanding has to come from our brains, not a machine's. So that initial feeling of being completely lost, I call that the paper panic problem. It's that moment where you feel like you're drowning in data you can't even begin to understand. But don't you worry, we're about to solve that for good. Welcome to your Scientist Survival Kit. This is it, a straightforward, step-by-step strategy that's going to help you tackle any paper with confidence. No more panic. From now on, just a clear plan of attack. So the heart of our strategy is something I call the 3-read rule. It's so simple, but it is incredibly effective. Your first read? That's just a quick skim. You're just getting the big picture from the title, abstract, and figures. The second read is where you really dig in and focus on what the scientist actually did, the methods and results. And that third read? That's when you get to analyze what it all means. This layered approach is key because it stops you from getting bogged down in tiny details too early on. Okay, so where do we start? Always with the abstract. Think of it like a movie trailer for the entire paper. In just a couple of sentences, it gives you the background, what they did, what they found, and the final takeaway. This is your first major checkpoint. It helps you decide if this paper is even relevant to you. If the abstract doesn't grab you, you can move on without wasting hours of your life. Next up is the introduction. This section is all about the why. It sets the stage, explains what we already know about a topic, and, this is the important part, what we don't know. You're basically hunting for the research gap. And here's a pro tip. It's almost always spelled out for you in the very last paragraph. That gap is the whole reason the study exists. Once you find it, you understand the one big question the entire paper is trying to answer. Now we're getting to the real heart of the paper, the methods and results. This is the how and the what. Here's my best advice. Don't just read the text here. Seriously, focus on the figures and the tables. They almost always tell the story way more clearly than the paragraphs around them. For each figure, just ask yourself, okay, what question is this trying to answer? And finally, we've got the discussion. This is the so what section. This is where the authors get to interpret their findings, compare them to other studies, and importantly, admit what their study's limitations were. And this is where you get to put on your critical thinking cap. Do you actually agree with their interpretation? Could there be another explanation for what they saw? Now, remember the Great Proposal Disaster? AI isn't the enemy here, not at all, but you have to use it the right way. So let's talk about how to use AI the smart way, as a powerful assistant to help you understand, not as a replacement for your own brain. I love this quote because it perfectly reframes how we should think about AI. Treat it like a super knowledgeable lab assistant. You can ask it to clarify a confusing sentence, explain a complex concept, or summarize a really dense paragraph. But at the end of the day, you are still the scientist in charge. And this shows you exactly what I mean. Asking an AI to summarize the entire paper for me, that's just lazy, and you're not going to learn anything. But asking it something targeted like, hey, can you explain the RT-qPCR method they used in this paper in really simple terms? See, that's a specific question that helps you build real understanding. You got to be specific, be curious. And here are a few tools to get you started. So if you just want the main ideas of a section, tools like Scholarcy or SciSummary are fantastic. If you want to ask specific questions about a figure right there in the PDF, you should try something like Explain Paper. And for just breaking down those really tough scientific concepts, Perplexity.ai and ChatGPT are basically the best lab assistants you could ask for. All right, theory is great and all, but let's put our new skills to the test. Let's get our hands dirty. We're going to apply our whole survival kit to a real world, cutting edge paper about a CRISPR diagnostic for COVID-19. Let's see how this all works in practice. Okay, here's our paper. That title looks a little intimidating, I know, but we have our strategy now. Let's break it down. FAACAS9, okay, that's some kind of CRISPR thing. CRISPR diagnostic, SARS-CoV-2 variants on a paper strip. See? We're already getting clues. It's a test for COVID variants, it uses a specific CRISPR protein, and it somehow works on a simple paper strip. Before we go any further, let's just quickly define a key term here, CRISPR-DX. That stands for CRISPR Diagnostics. It's this really revolutionary new method that takes the power of CRISPR gene editing tools, in this case a protein called Cas9, and uses it to detect diseases and genetic mutations really, really quickly. So let's do our first read and just tackle the abstract. In just a couple of lines, we get the entire core idea. Scientists took a super specific CRISPR protein, FNCas9, and they repurposed it to build a simple paper strip test. And what's the goal? To detect not just the SARS-CoV-2 virus, but also a really critical mutation called N501Y, and to do it all in under an hour. See, that's the whole story in a nutshell. Reading that abstract leads us to a really natural question, which is exactly what a good reader should be doing. The paper calls their test RAY, which stands for Rapid Variant Assay. So how does RAY actually work on a paper strip? This question is what pushes us from the what of the abstract to the how of the method section. Okay, let's break this method down. As you can see from these 4 steps, it's actually pretty clever. First, they take a sample from a patient and make a ton of copies of the virus's genetic material, its RNA, and they tag it with a little molecule called biotin, which basically acts like a tiny sticky flag. Second, they mix all of that with a custom-designed CRISPR complex that's been programmed to hunt for that specific viral RNA. Third, they just dip a simple paper strip into the mix. And finally, and this is the cool part, if the virus's genetic code is there, the whole complex sticks to the strip and a visible line appears. It works just like a pregnancy test. So, did it work? What were the results? Well, here's a huge data point. The RAY test correctly identified the N501Y mutation with 87% sensitivity. That means it caught 87% of the true positives. And it had 97% specificity, which means it correctly ruled out the mutation in 97% of the negative cases. For a rapid, simple paper strip test, that's a really powerful result. So, there you have it. We learned the strategy, and we've already applied it. Let's wrap everything up with a final checklist that you can use to unlock the secrets of any scientific paper you ever come across. Okay, here it is, your final checklist. First, always identify the main research question. What's the point of this whole paper? Second, be critical. Understand the techniques they used for sure, but also keep an eye out for any weaknesses or maybe missing controls. Third, think about the big picture. How do these findings actually fit into the wider scientific field? And finally, like we've been saying, focus on the figures. If you can explain the figures to someone else, you've really, truly understood the paper. This is how you start to think like a scientist. And just like that, the world of scientific research isn't locked behind some sperry wall of jargon anymore. You have the keys, you have the map, and you've got the tools to explore it. So the only question left is, what are you going to discover next?